Thank you. Focusing on how we can characterize the tumor microenvironment using multiplex immunofluorescence, especially utilizing AI-enhanced spatial image analysis to do so. And we will focus on characterizing proteins. The tumor microenvironment is a quite complex structure. There are lots of different players, and this complexity drives the need to also have a technology being able to access all these different cell types. In histopathology, I mean, we come from a more architecture-focused diagnostics on H&E on the left, and then started to utilize antibodies to characterize proteins to identify cells, status like proliferation. It's also used as a predictive biomarker like for PD-L1 and HER2. And with immunofluorescence, we can bring this to the next level on proteins by visualizing multiple proteins on the same section, giving access to more cell types or cell subtypes, including their spatial organization. And in this area, Ultivue developed two technologies that we offer to enable faster, high-scale, and robust and reproducible science there. On one end, we have our assay technology. It's easy to use, modular, and really meant for large-scale and high-throughput analysis. I will quickly explain it, but then really focus on the analysis part, because we think MIF not just as an assay plus analysis, but like as an analyzed assay, and therefore developed a very targeted image analysis that also focuses on reproducibility, automation, and really scalability to remove this bottleneck that we already saw today. It's a lot of data we generate, it's complex data, and we need the targeted tools to really overcome this. Yeah, therefore, we really think that as an end-to-end approach from samples to insights, where we want to assist. And the first step is our assay technology. It focuses on up to 12 targets, really meant to run on many samples on whole slides. And to quickly introduce you on how it works, it starts with the deoxin antigen retrieval step. It runs on FFPE sections, and then we have one single antibody binding step. So it's not a sequential staining. We use sequential imaging, but the staining is actually one step where we deposit and bind all antibodies at the same time. We just show 8, but it's up to 12 at the moment. Comes as ready-to-use kits, or we can also support as a service lab. The antibodies are conjugated to small DNA strands. We call them barcodes, that are designed carefully in a way that they're complementary to each other, so they don't interact. They have minimal, unspecific binding to the tissue. And then what's also important as a next step, we amplify these barcodes, which means they result in having then multiple copies, which then in the end also amplifies the signal in a way. So that, for instance, our signal is so strong that we don't need to remove autofluorescence when analyzing the images. And then the actual readout phase starts, and that one is cyclic. We on purpose always go in rounds of 4, and the idea was to enable a multitude of different scanners that support like 5 colors, and then to also avoid any computational spectral unmixing steps that can also be complex. And here then, we add 4 probes with 4 different colors, do an imaging step. This is followed by a very gentle dehybridization step where these probes are removed after the scan, and then the next round starts by depositing the next probe mix and doing the next scan. And again, that's a gentle process. At the moment, we routinely offer it 3 rounds, and then the samples can still be used for further analysis. We like to do an H&E to bridge like the molecular focused multiplex immunofluorescence with classical pathology, but also other techniques are possible. Which then brings us to the next step that can be challenging. You have multiple scans and you have to put them together, ideally in a subcellular resolution, so that you can combine the markers coming from different rounds of imaging, so that you can really do deep phenotyping and excellent colocalization. And this can also be computationally quite challenging. Our technology is always applied on the whole slide, so big resections can have millions of cells, and therefore we invested a lot in having highly scalable solutions. And the next one is our co-registration software, Ultistacker.ai, that then can co-register the different IF scans, plus, for instance, an H&E or other modalities. And to show some details, yeah, an example image where we have an 8-plex plus an H&E, where the channels are shown on the right. And I only visualize the two MIF DAPI rounds, one in blue and one in yellow. And since they overlap, we see white. So it's not one single DAPI, it's actually two DAPIs that we're seeing. So you see that 3 is subcellular resolution that we achieve. It not only works on this nice image, it also works when artifacts are present. I mean, real life data can look very different. So the algorithm is not disturbed by tissue loss or some very bright DAPI artifacts from dust or reflections. The algorithm runs scalable in the cloud, so we can run hundreds of images in parallel. We only need to upload images to a cloud repository, and then can get back the registered image within a few hours. As said, you can also add an H&E to our IF stack, and for registering that one, it's the same section, so same architecture, so you want to have it at the same level of quality, can also be challenging. Here we wanted to remove any manual interaction, since that's like the key to automation. And therefore we are utilizing an AI method, a generative AI method called generative adversarial networks, where we trained this model to predict a DAPI signal from an H&E. And here you see an example from our training set. So you have the H&E section we had with a more traditional method co-registered already an IF DAPI, and then predict from the H&E the DAPI. And you can appreciate it's extremely similar. And that allows us to then transform the H&E into a DAPI representation and co-register it similar to the previous one, which then removes any manual settings. And finally, here then overlaying the H&E with the first DAPI channel in bright green, and appreciate that the nuclei again are exactly overlaid, and the algorithm is not confounded by some tissue differences or some blurry regions, as noted here with the notations. So once we have all these markers and channels overlaid, we can look at the cells and really how the assay stains them. And in this case, just an example, we see that even in the same cell type, we can have a multitude of different expressions that then are overlaid. So we can combine them then all into a deeper phenotype. And that shows why it's so important to have an excellent co-registration on this single cell level to really be able to combine these. If there's a small shift, you would then have a false colocalization of staining. It also shows that this data can become really complex. So coming from like IHC, where like a human reader is still key, on MIF, you really need to have computational tools to read such an image. There are millions of cells on it, with colocalizing expressions, and it's only possible via a computational approach to make sense of this complex data. And this brings me then to our next step in our image data science pipeline. We call it STARVUE for Spatial Tissue Analytics and Reporting on our view panels. And again, after the stacking, we have developed a very targeted image analysis approach, and then this is followed by the actual data mining or insight generation, where you then combine your MIF-based spatial data with other modalities, with treatment information, with response information to actually generate the insights that we already learned today. For our image analysis, we take as input the previously co-registered image. H&E is optional. And here I have to say we really chose to use deep learning for every step in the image analysis process. So not just the cell segmentation, but really every step. And it's not because AI is fancy, but it really helps you to adapt to this huge variability that we face on tissue. And I started in classical image analysis like more than 10 years ago, and a lot has changed. Now we have very powerful tools. Most of them were developed on natural images. And here it's really key to select and transfer these tools to our IF domain. And the first step we do is a tissue segmentation to define the tissue on the section. We always analyze the whole tissue, not leaving out any data, and use a semantic segmentation to distinguish between foreground and background at low magnification and DAPI. And then as a second step, we do another semantic segmentation to identify regions. Our panels are often used to study solid tumors. We often have cytokeratin as part of the panel and utilize that to do then a tumor versus tumor stroma separation within the actual tumor region. And here an example, again, a second semantic segmentation approach. And then this is followed by a cell segmentation. We already saw that in previous talks. I mean, this is an instance segmentation, really finding every instance of a nucleus done on DAPI on high magnification. I would say until now that's all pretty standard in the current approaches. What is really remarkable and most differentiating is the next step, where we classify the positivity for every marker in the panel. And we do that not based on thresholds measured like within these cell objects, but we actually, again, train a deep learning method. We choose vision transformer-based object detection, really a state-of-the-art, also coming from the natural images space, and train in a supervised way by annotating many cells in our own image data to then predict the cell position and outline in such a box, like bounding box way. And this is done for every marker independently, because also our panels are put together independently or in a modular way. And then this can be mapped back to the previously segmented nuclei, really generating then a feature vector per cell where you have marker positivity or negativity. This whole data is combined into this one data structure and then can be refined for further reporting with manual annotations, either selecting regions of interest or masking, like a tumor versus benign or some artifacts. Importantly, this can be done at the end without redoing the whole analysis step. And also the phenotyping is just then filtering the data set for the combination of markers that you're interested in, so you can dive into all kinds of subpopulations at will. A few slides to the actual performance of our deep learning approach, especially this marker positivity detection. We use AI here again to be able to automate this whole analysis process, so reduce the manual engineering of thresholds, for instance. We have algorithms that can really adapt to a huge variability, so they can the same algorithm can run on large clinical trials, being it multi-year ones. The deep learning models improve over time. I mean, we can add training data, so it's really a continuous improvement process. And we bring like pre-trained models trained on our like core library of markers, and then our own data, so that it's really fast tracking the whole analysis process. Just some more visual examples. Here we show PD-1 in red that can be a high or low expressor, and therefore it's a good way to show that the same model can really detect both extremes. Sometimes markers can have a multitude of different morphologies, and also here the models can adapt to them. On top, important to note that the prediction done by the object detection not only looks within the cell, but really also outside. So it looks at the context. is there any, is it a clustered region? Is it the sparse region? Do I have some unspecific staining surrounding it that can confound my positivity assessment? And this is trained into the whole system, which then really leads to robust and high accurate data. We not only rely on nice images, we also follow good practice in machine learning development. So in addition to our training data set, we also annotated, or our pathologists annotated an unseen validation data set. This is also continuously being extended, done for our core marker library. And here I show on the left the scatter plot where we compare the counts per annotated field of view against the predicted count. We see that every dot is a field of view, color-coded by the marker it was annotated for. And we see that it's aligning on this dashed line, which would be perfect agreement. And this is also represented in the metrics. We like to use Linz concordance coefficient, correlation coefficient, which compares the two counts per field of view and goes from -1 to 1, 1 being perfectly concordant. And here again, the overall Linz correlation coefficient is at 0.95, and every single marker one is above 0.8. And also our F1 scores are also then looking at the actual position of the cells on top are really high, showing that we have very accurate detection. Of course, it's deep learning. I mean, this is tested on a certain data representation. If the data we want to analyze is very different, we have to extend the training data and also the testing data to incorporate that one. Yeah, finally, once you build trust in your analysis, you have all the cells, then the actual spatial analysis starts. I won't go into a lot of details today, but the typical approach we choose is to take all the cells, put them in such a graph representation. So every dot is the cell position, the color encodes the phenotype, and the neighbors are identified just by a radius surrounding them and connected with lines. And then you can start to pose different questions, as we heard earlier today. You can look at neighborhoods. Is cell A and B in the neighborhood? You can start asking questions like, can the immune system attack? So for instance, let's count the number of tumor cells that are close to a cytotoxic T cell. Or you can look at infiltration, as shown in the bottom, where for two cell types, we are looking at their position, the relative position to the tumor and tumor stroma border coming from our segmentation. And here for the yellow cytotoxic T cell, we see within the tumor this shoulder, like indicating infiltration when compared to the helper cells. This again is done on all cells, a full slide, so can be millions on one slide, but also across cohorts. So it's really targeting large numbers of cells here. And is also, in our case, computationally accelerated by cloud computing. This closes our end-to-end approach. Of course, the final step is then the scientifically most valuable one, where you combine the spatial readouts with additional information, being a treatment information, response information, and where Ultivue can help to really fast-track your science to go there by avoiding some tedious, often tedious assay and panel development or image analysis development steps. Yeah, before closing, I would like to show one example of such an end-to-end approach that our service did in a routine project where we built a 12-plex, the following 12-plex for a customer, and then on top did a verification study where we looked at the reproducibility of this assay, because especially if we think of multi-year trials, it's really key that every time you run a new batch of analysis, you have to be sure that you have reproducible data. So yeah, that's a panel. I won't read all the markers, and that's experimental design. So we analyzed 9 consecutive sections where we had an intraday and interday test always 3 sections per day. The consecutive sections were distributed in interweaved way across the days to reduce like the biological variability or account for that one. And then we stained, scanned, and analyzed with the same settings or algorithms all sections. We addressed the reproducibility by computing the coefficient of variance for the mean signal intensity computed on all cells, and also the cell density over all detected cells. And for our assessment criteria, we chose that staining that is within 20% or less of the CV, we treat that as an acceptable range of variability. First, let's look at the raw data across all samples. On the left, we show the mean positive signal intensity. Again, every dot is one of the sections, and we have all the markers, and we can already appreciate that the dots are rather clustered. And on the right, even more clustered, we have the positive cell density, which is the actual readout you would use in your spatial analysis downstream. And looking at the CVs coming from that data now, including the intra and interday separation, for all markers, we are way below our 20% target for both the raw intensity that you would use in manual intensity-based gating, but also in the positive cells coming from the deep learning classification, where we have the highest CV for FOXP3 in the interday, around 15%. Yeah, so in summary, to close, my key points were first, MIF in a flexible and modular way is key to study the complexity of the tumor microenvironment. It allows us to study subpopulations, since we're looking at the same section, to really dive into the spatial organization. Here, Ultivue's InSituPlex method offers easy to use, highly sensitive, customizable, and mostly reproducible way to do that. It's not restricted to any specialized hardware. It can run on an auto stainer. Also, the way we do the readout with the 5 colors does not restrict it to a certain scanner and can be used with a multitude of ones. Of course, you generate a lot of data, very large data, so you have to overcome the complexity and the size challenge, and you need specialized tools to do so. And here we developed STARVUE, our analysis platform, which is highly scalable, based on cloud computing. In my opinion, the only way you can really solve that, and fully deep learning enabled, so that you can really run, once you have developed it, your analysis in a very automated and unbiased way, adapting to the huge variability that you get on tissue. And yeah, finally, applying such an end-to-end approach, so thinking the analyzed assay, here our 12-plex data shows that on the quantitative cellular data could achieve excellent reproducibility. Thank you.